Hi everybody, my name is Karthik and today I will be discussing about fetal rubella syndrome, also known as congenital rubella syndrome. To start off, rubella virus is a viral infection similar to measles and it's distinctively known to cause a red rash on those that are infected. It also causes excessive inflammation around the body and that could sometimes cause the adults to develop arthritis. However, in fetuses, it is much different. The rubella virus causes many birth defects, such as deafness, with the most common defects found in the eyes, hearts, and the brain. Symptoms can often be cataracts, microcephaly, and many heart problems that could last throughout a lifetime. According to the World Health Organization, there is a 90% chance of the fetus contracting the virus from an infected mother. Sometimes the pregnancy leads to an unfortunate miscarriage situation. The direct effect of the virus is unknown, but what is known is that the timing of the infection matters. If the fetus is infected early on in the gestation, around the 12-week period, the fetus is likely to develop many birth defects, whereas after the 20-week mark gestation period, there are rarely any defects documented. Currently, there are no antiviral treatments to combat the rubella virus infection, let alone during a pregnancy. The only preventative measure is to take the rubella vaccine before becoming pregnant. And then from there on, it really depends on the infection and the birth defects the baby was born with for the respective treatment that could possibly help alleviate pain and prolong life. So for my presentation, I will be reviewing and analyzing this paper. This paper looks into how the virus impacts development early on using human stem cells. The purpose of the study was to understand more about the rubella virus and how exactly it causes trouble during early embryogenesis. Also to why the effects are greater early on in the development. There is a 90% chance of the virus being transmitted to the fetus, with the transmission via shared blood supply. This study builds upon a prior study where the effects caused by the rubella viruses were experimented on mice. As we learned in class, mice are a model species that have similar embryonic development to humans, including similar homologous genes and transcription factors. That study concluded that the virus affects the three germlines found in developing embryos differently, and the endoderm cells were affected the most. This study now followed through to determine whether or not this is the same in humans, whether or not the endoderm cells were affected more. The main obstacle of this experiment was finding an ideal variable to test with. Since it is unethical to use humans in the study, the authors used human pluripotent stem cells. These cells were used because they can maintain their pluripotent property and identity after the differentiation process. I think this is an ideal choice. These cells have the ability to differentiate into almost all types of cells, much like the embryonic stem cells during development. The authors mentioned that they did not use embryonic stem cells because pluripotent stem cells were much easier to grow into embryoids in terms of toxicology. Embryoids are three-dimensional aggregate structures of stem cells. The stem cells are ideal also because they retain the human genome. That way, we can observe how the virus interacts with human genes. As for the control variable, the authors used a lung epithelial cell line of a carcinoma patient. The paper did not include why a carcinoma cell line was chosen as a control. I infer that much like stem cells, cancerous cells exhibit the proliferative signals to help continue their metastasis. Compared to other somatic cells, cancer cells are far more similar to stem cells in that manner. They keep proliferating. The cell cultures were then exposed to the rubella virus sample, which was derived from a monkey epithelial tissue collection. After incubation with the virus, some of the pluripotent stem cells were induced with differentiation signals, and some were not induced. This was done to observe if the virus had an impact on differentiation early on, even before germ layers were determined. The pluripotent stem cell cultures, regardless if directed to differentiate or not, produced the three germ layers, endoderm, mesoderm, and ectoderm. The cultures were then grown into embryoid structures. The sequences of the viral RNA was then used to set up about 48,000 gene probes that was used to microarray the viral and cellular RNA in the embryoids. A quantitative real-time PCR process allowed for quantification of the viral mRNA as well as the cellular mRNA that are necessary for embryo development. The viral mRNA was also exposed to fluorescently labeled antibodies to track their positions in the embryoid in reference to other genes. In terms of how the experiment was designed, I think the experiment would have been more representative of the problem it seeks out for if the experiment used embryonic stem cells. I'm also not sure about the use of a carcinoma cell line, especially as the control. I understand its proliferation properties make it an ideal contrastive option to stem cells, but as a control, I do not think it is ideal. Cancers are very unique and genetically different from one another. A possible difference in mutation of the carcinoma cell line may mean it can interact with the viral content in an unprecedented way, thus introducing unnecessary variability into the experiment. The experiment also only considered one replicate with a positive control. The study could have included mature somatic cells from tissues that would have been affected by the rubella virus during embryogenesis. This would show how the mature cells are different in that the rubella virus does not affect it like it does in the early predecessor version of that tissue. It is worth mentioning that it is appreciative that the experiment considered to observe a broad range of genes, focusing on the ones that do cause the birth defects. I believe that broader options are more representative of what occurs during an infection, positive potential correlation. I would also suggest the use of more replicates of appropriate cell types and using embryonic stem cells as the control instead. All in all, the study was able to produce interesting findings. The key findings of this experiment were that rubella virus has an effect on cell adhesion. It was observed that the infected cells were unable to cluster together when compared to the normal embryoids. Rubella virus also was seen to affect the three different germ layers differently and affects the endoderm the most. Rubella virus does not affect differentiation of the cells when they're becoming the ectoderm, mesoderm, or endoderm tissues, but afterwards when the cell begins to adopt a specific identity from there on. The rubella virus also activates the IFN response signaling pathway, which was also observed in influenza virus infections and pneumonia infections. Focusing in on endodermal cells, it was observed that the TGF-beta signaling pathway was reduced effectively with the introduction of rubella virus. This figure shows the prominent genes that were manipulated and how it results in the different birth defects during a rubella virus infection. Rubella virus was seen to affect ectoderm cells by downregulating the expression of SIX3, which is responsible for the development of the eye field. Rubella virus affects the mesoderm by downregulating Wnt and HAND1 signaling. The changes in the expression within the ectoderm and mesoderm were not as significant as the changes in the endoderm cells. Of many transcription factors, such as SOX17 and CER1, many were upregulated and downregulated, and this caused disruptions in the cell's ability to maintain its identity and differentiate any further. This figure shows the process data of the microarray analysis of some of the developmental genes present in the embryoids and their interaction with the rubella virus. Similar to the previous figure, this figure shows the prominent genes that are necessary for the development of certain tissues that are impacted by the rubella virus. Towards the left, you can see there is very little expression through the lack of color or lighter gradient. Blue indicates a downregulation and the orange-red indicates an upregulation of certain genes in the presence of the rubella virus. The conclusion. The purpose of the study was to observe whether the rubella virus affected endoderm cells the most in humans. The experiment failed to falsify the hypothesis as the results showed that there is change caused by the rubella virus in all three germ layers of an embryoid, and of the three, the endodermal cells were influenced the most. The study was also able to support the idea that rubella virus affects embryogenesis in another way as well, through cell adhesion. The authors of the study infer that this is the result of a down-regulated Wnt signaling. The authors made it clear that this is only the beginning. Now that their hypothesis is supported, future research will look into how and why rubella virus interacts with endodermal cells especially. This was an interesting topic to learn about, especially the fact how the virus affects an embryo far more detrimentally than it affects an adult. I am left intrigued more about the virology and embryogenesis, and I'm left with more questions. Why does rubella virus affect endodermal cell pathways specifically? How do the viral mRNA and proteins physically interact with the signaling molecules of the cells? And can pharmaceutical drugs be formulated and synthesized that could counter-combat the viral proteins and possibly stave off an infection? That is all for my presentation. Thank you.